Hello children, welcome to biology class. Today let's start the class by explaining the salient features of the double helix structure of DNA. Last day we discussed about the structure of DNA. Even though we can discuss it again, what is the first feature? It is made up of two polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. So here you can see the nitrogen bases, then deoxyribose sugar and a phosphate group. Okay. So which are the four nitrogen bases found in DNA? Guanine, cytosine, adenine, thymine. Isn't it? Then the two chains have anti-parallel polarity. It means, see here, one chain has the polarity from 5' to 3' and the other one has polarity from 3' to 5'. That's why both the chains have anti-parallel polarity. Clear? Then the third feature, the bases in the two strands are paired through hydrogen bond. See, between thymine and adenine and guanine and cytosine there are hydrogen bonds. Here it is double bonded. That means between thymine and adenine, two bonds. You can see double bond. Whereas here three bonds, three hydrogen bonds. Adenine forms two hydrogen bond with thymine. See, it is clear from this picture. Two hydrogen bonds. And thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with three hydrogen bonds. Clear? As a result, always a purine comes opposite to a pyrimidine. This generates approximately uniform distance between the two strands of the helix. See, one purine always bond with one pyrimidine. That's why approximately uniform distance is there between the two strands of the helix. Then the fourth feature, the two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer. And there are roughly 10 base pairs in each turn. See, the pitch of the helix is 3.4 nanometer and there are roughly 10 base pairs in each turn. See, here you can see, here right is a right-handed helix. In each rise 0.33 nanometer nucleotides, pitch is 3.4 nanometer, 10.4 nucleotides per turn. Here 10 pairs of 10.4 nucleotides per turn, and here you can see two grooves. This is minor groove and this is major groove. Okay. Then the distance between base pairs in the helix is equal to 0.34 nanometer. That is 3.4 divided by 10. 10 nucleotides are there now in one turn. Clear the calculations? The plane, then the fifth feature, the plane of one base pair stacks over the other in double helix. Thus, this in addition to hydrogen bond confers the stability of the helical structure. Okay, children? The proportion of the double helix structure for DNA and its simplicity in explaining the genetic implication became revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from the DNA to RNA, then to protein. The DNA undergoes a process known as transcription, and a single-stranded RNA is formed. It undergoes another process, translation, and proteins are formed. This is central dogma of molecular biology. Clear? So, the DNA is unwound by the process of replication, and by undergoing a process transcription, it is converted to RNA, and it undergoes translation, it becomes protein. Okay? This process is termed as central dogma of molecular biology. This process, replication, transcription, translation, we will discuss later. But you keep it in mind, this is central dogma of molecular biology. But children, in some viruses, the flow of information is in reverse direction. That is from RNA to DNA. And this is reverse transcription, RNA to DNA. And this process is happening with the help of enzyme reverse transcriptase. Okay. Next, we are going to discuss another topic, that is packaging of DNA helix. Children, why this packaging is required? I will explain a calculation for you. Children, listen. Taken the distance between two consecutive base pair as 0.34 nanometer, if the length of DNA double helix in a typical mammalian cell is calculated, it comes out to be approximately 2.2 meters. That means a length that is far greater than the dimension of a typical nucleus, because it is having approximately 10 to 6 meter. Okay. Then, how we got this calculation? How we get this 2.2 meters? Children, you just multiply the total number of base pairs with the distance between two consecutive base pairs. That is 6.6 into 10 raised to 9 base pairs into 0.34 into 10 raised to minus 9 base pairs. So, we'll get the answer as 2.2 meters. So, this much big DNA has to be included within the nucleus. Isn't it? See, it is having 2.2 meters. So, here comes the importance of packaging. So, how such a long polymer is packed in a cell? That we are going to explain now. So, first of all, we can explain the packaging of DNA in prokaryote. Then we can explain the same in eukaryote. Children, in prokaryotes such as E. coli, though they do not have a definite nucleus, the DNA is not scattered throughout the cell. Instead, the DNA, we know that it is, yes, negatively charged, is held with some proteins with positive charge in a region termed as nucleoid. So, the DNA's nucleoid is organized in large loops held by proteins. See, here the DNA is loop and protein, positively charged protein. Clear? The DNA packaging in prokaryotes. Now we are going to discuss the DNA packaging in eukaryotes. In eukaryotes is much more complex, children. This packaging is much more complex. There is a set of positively charged basic proteins called histones. See, here histones are given in a purple color. Histones. This protein acquires charge depending upon the abundance of amino acid residues with charged side chains. Isn't it, children? I think we discussed the structure of protein in class 11. Isn't it? Amino acid. It is a polypeptide. Amino acids are connected by CONH bond. The protein is produced. So, these histones are rich in basic amino acid residues, lysine and arginine. That's why it is positively charged. You listen the diagram too. Okay, here you can see DNA wrapped around the histone. Can you see? Then both the amino acid residues carry positive charges in their side chain. That's why histones will be having a positive charge. And one more speciality is therefore histone. They organize to form a unit of 8 molecule called histone octamer. The diagram is there in our textbook. H3, H4, H2A, H2B, and here one H1 also. Okay, and it is termed as histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome, and this model is explained as nucleosome. So the question will ask to explain the nucleosome model of DNA. So a typical nucleosome contains 200 base pairs of DNA helix. And this nucleosome constitutes the repeating unit of a structure in nucleus called chromatin thread-like stained colored bodies seen in nucleus. Okay. So here you can see linker DNA to connect between two histone bodies, and this in red color it is core DNA, and this is in blue color it is histone octamer. Which are the units? H2A, H2B, H3, H4 into 2. Na? On both the sides there are histones. Clear? Then the nucleosome and chromatin are seen as bead on string. That's why this nucleosome model is also explained as bead on string model structure. This is a view on an electron microscope. Here you can see this octamer, histone octamers as bead and the DNA as string. The beads on string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosomes. And the packaging of chromatin at higher level requires an additional set of proteins that collectively referred as non-histone proteins. See, in green color you can see an additional set of proteins, that is non-histone chromosomal protein. Children, in a typical nucleus, some region of chromatin are loosely packed. See, it is termed as euchromatin and are referred as euchromatin, and a chromatin that is more densely packed and stains dark are called heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Okay, so that's about packaging of DNA. Children, you read the textbook thoroughly. Next day we can explain different experiments put forward by different scientists to find out the genetic material as DNA, and how they analyze or how they conclude that DNA is the genetic material. So we can explain those experiments on next week. Okay, so today you study the textbook thoroughly. Okay, thank you children.